So Ceecee is a Senior Research Fellow and Group Leader in the Ocular Genetic Research Program and VENTURE Victorian IRD Registry at the University of Melbourne and Centre for Eye Research Australia. And she leads amazing research to improve the diagnosis and understanding of inherited retinal diseases. Today she'll be providing an overview of genetic testing for IRDs and why it matters. Thank you, Ceecee.
Thanks, Eden. Hi, everyone. I've had the pleasure of meeting many of you over the last few years. And if I haven't, then do please come and say hi later. But thank you for having me, and thank you very much for coming. I really hope today is a great opportunity for everyone to connect, both with us and with each other, because I really do think we can make a huge difference. I'm Ceecee, as Eden mentioned, and I lead a lot of the ocular genetics studies at the University of Melbourne and Centre for Eye Research Australia. Now, before I start, I know the mic sounds a little bit echoey, so if anyone can't hear me, just wave your hand and I'll try and slow down or enunciate better. What I'm going to talk about for the next 15 minutes is really the journey of diagnosis. Not only getting an inherited retinal disease diagnosis, but also where genetic testing might fit in as a part of that journey, as well as other components like finding support and also being involved in research and clinical trials. We know that diagnosis for many inherited retinal diseases often start with an eye test. Whether or not you have any familiarity with IRDs, because you may have someone in the family or know someone with it, or no familiarity at all, that's often I know everyone here probably has their own journey and their own story to tell. You may be along different parts of that journey, and somewhere down the road, genetic testing may also fit in as a part of it. Then along the process, it's also important about finding the right support. And I'm not only talking about practical support, so things like occupational support, vision aids or guide dogs when that becomes relevant, but also emotional support, finding peer support, finding groups like Retina Australia that may help you, as well as them participating in research and clinical trials so that we can all work together to really improve support and care for inherited retinal diseases. One thing that we really wanted to understand a few years ago is actually what is the diagnostic journey like for people with inherited retinal diseases, especially at the earlier stage of diagnosis. And so we undertook a study where we asked a lot of people with inherited retinal diseases, actually, what was that journey like, or what is that like for you, and what are some of the challenges that you've had as a part of it? And I know some of you in the audience would have been a part of that study, and we're very, very grateful to have heard your stories. But what we really found is that that journey can be very challenging. And some of the aspects that make it challenging is because inherited retinal diseases are progressive conditions. And so the story can change alongside of that journey. We found that among different people with inherited retinal diseases, some of the common themes that we've heard include adapting to the diagnosis can be very difficult, because for many people, actually, they don't know anyone with inherited retinal diseases. So getting a diagnosis can be a bit of a shock, and not understanding sort of what that might look like in the future. We also then looked at the practical challenges. So we know that there are challenges with navigating different parts of daily life, both in terms of reading and in terms of navigation, and finding support, I think, in schools and in workplaces. But also something else that we wanted to recognize is that it's a roller coaster of emotions that many people reported that they do go through living with inherited retinal diseases. And that's managing the mental load of having to think ahead, of having to plan ahead. We also found that many people found navigating society or different parts of society difficult because inherited retinal diseases are not always visible, and it is a hidden disability. And lastly, we heard a lot of stories about really a changing identity that comes with the diagnosis, and also many positives that came with it about how people have really found resilience and have managed sort of that changing identity and found a way forward. And really what we wanted to do with the study is really highlight the experiences that different people with inherited retinal diseases have. And so we can really come together and figure out how we can better provide support along different parts of that journey going forward, and leading into research and new treatments for these conditions. But really what I'm going to talk about today is also where genetics fit in. So why is it important that we have genetic information and a genetic diagnosis in inherited retinal diseases. So like Lauren said, inherited retinal diseases are generally caused by a single gene. So there's a single genetic cause in most people who have an IRD. Therefore, there's many personal benefits to actually finding out what that cause is. And I'm not going to go into the specifics of the personal benefits because Parker's going to talk about them next, but we know that it can help us really confirm the diagnosis and give us clarity. It can give us information for family planning and for other family members as well, now and into the future. And also finding out the specific gene that's causing an inherited retinal disease can help us get people into specific treatments and clinical trials for their condition. But outside of these personal benefits, actually knowing the genetic diagnosis helps us further research because it helps us understand the different disease mechanisms that are associated with each inherited retinal disease. And therefore, that helps us develop and also design better clinical trials that can target these conditions. But the process of genetic testing is not always as simple as submitting a blood sample, going to the lab, and then having the machine spit out a report. It's a little more nuanced than that. And I'm going to talk a little more about actually what is that process that we do go through. And so often what happens is after you submit a blood sample, the blood sample goes to the lab and it gets sequenced to start with. And the process of sequencing is really chopping up your DNA into little bits and then putting it through a machine. That generates a lot of information. So for context, we each have 3 billion base pairs of letters in our body. And pretty much if you printed out all of these letters, that comes to about 3,000 phone books. And that is the amount of information that we actually process through each genetic test. After it gets sequenced, the lab often uses an algorithm to try and map it to a reference to try and find out where the genetic changes are in that genome. Because we've got to find the changes in order to find the one single change in that 3 billion that's causing an inherited retinal disease. The third part is then assessing the evidence for these changes. And that's what I actually consider to be really, really important. And I always say that when you get a clinical genetic test, in other words, when you go and see a doctor or a geneticist or a genetic counselor, and they do a test for you to find out what change is causing your inherited retinal disease, what they're really looking for are changes that have previously been reported and validated. In other words, when you do a clinical genetic test, what we're looking for is an already existing change, one that has already been found in another family around the world with an inherited retinal disease, and one that we have already proven to cause an inherited retinal disease. If such a change is found, then a report is generated and a genetic diagnosis is given. However, we know that currently only about 50 to 60% of cases can actually get a genetic diagnosis from clinical testing. What that means is that only about 6 out of 10 people have a change that's already been discovered. For the other 4 out of 10, it doesn't mean that they don't have an inherited eye disease. It just means that they've got a change that we haven't found yet, and we really do need to, and we want to as well. And it might not have been found because of testing limitations, for example. So the testing techniques, I've got to say, are actually really, really good now. But sometimes there are really tricky changes in the DNA that are hard to detect with standard techniques. It could be because of unknown DNA changes. And they're what we call variants of uncertain significance, where we found a change in the DNA, but there's 1 million other changes. And so we don't really know what this particular one means. And a third reason, it could be a change that we just have not found and have not validated to be associated to cause an inherited retinal disease yet. And so what can we do in these situations? What can we do for the 4 out of 10 people who can't get a genetic diagnosis from clinical testing? That's where a lot of our research comes in. And so with research, what we're aiming to do is look for new genetic changes, both by using newer sequencing techniques or by looking for other families with the same change so we can confirm what they might mean in terms of inherited retinal diseases. There are several different ways that we can do this, and it really depends, and it really is a case-by-case basis as to what might be the most suitable way. Sometimes we will test other family members, because if you imagine if you've got two people that are related, if both of them have the change, but only one of them has an inherited retinal disease, then it probably means that that change is not the one that's causing the inherited retinal disease, because it's also present in people who are unaffected. Sometimes we do lab experiments, and that might involve taking some blood, some cheek cells, nose swab, or sometimes a skin biopsy, where we can try and grow the cells in a lab to try and recapitulate that same genetic change to find out what it means. This doesn't always give us the answer because the changes that we see in cells on a plate and sometimes even in animals don't always sort of do the same thing in humans and in the human body. Sometimes we use newer sequencing techniques, and so that's something that our team is actually currently doing, looking at whether or not newer sequencing techniques can give us better and also more accurate answers. And lastly, and this is such an important one, is that we work with other teams. Because one thing that I think I've said is in order to validate new changes, we have to find at least one or two other families around the world with the same genetic change, with the same inherited retinal disease. And so collaboration is so important, because sometimes it really is like looking at a needle in a haystack. And so we work very closely with other teams, both in Australia and around the world, to work together to find these new answers for inherited retinal diseases. And what's next, really? What is our team doing? What are we really doing? Something that I really want to highlight today is, I think, a lot of hope that I've got for this field. I think around the world, there are more people working on research in inherited retinal diseases than there had ever been before. And there are some really smart people in this field. Particularly in Australia, our team works very closely with everyone, actually, all of the IRD experts around the whole country. We collaborate very closely with Sydney, with Perth, with Queensland. And so we have a wonderful team really working together to try and progress this field, try and develop new treatments, and to try and bring new treatments into Australia. And what we're really working on is first finding more answers. Particularly in our team, we're looking for finding more answers for people with inherited retinal diseases to get a diagnosis, and finding more answers in understanding how and why different inherited retinal diseases progress differently. We're also working on improving the diagnosis journey, so both genetic and clinical diagnosis, getting a genetic diagnosis faster and more efficiently, and supporting people in their ongoing journey. And lastly, many of you would have been invited in to participate in different vision testing studies. And that's also really important as well, because something else that we're working on is try and understand inherited retinal diseases better so that we can better design clinical trials. If you imagine if we've got 100 people with the same inherited retinal disease caused by the same gene, then we can learn more about that particular disease. And therefore, we're more likely to be able to develop treatments for that particular condition. And also you can develop the best treatment, but if you can't figure out a way to measure how that treatment works and show how that treatment works, then it's going to be really hard to get new treatments through regulatory approval. And so that's something that we're working on, is understanding more about inherited retinal diseases, how they progress, what are the best ways that we can measure treatments, therefore to guide more treatments in clinical trials and bring them over to Australia. And lastly, our team can always be contacted with the email ird@groups.unimelb.edu.au. Many of you probably will have the email already. And I'm going to finish by really acknowledging the wonderful team in Melbourne who have worked on all of these studies, both the genetic studies and the VENTURE studies, some of whom will be here today. And do get in touch with our team or come and chat to me after this if you've got any questions.